All right, so let's get right into incomplete dominance and codominance. When we use the term incomplete dominance, what that means is a heterozygous individual will look different than either of the homozygous. So, homozygous dominant and homozygous, so recessive, look different from the heterozygous. Nope. So each has their own pheno. So normally in complete dominance, which we've talked about before, the homozygous dominant and the heterozygous, that was weird, have the same phenotype. But now we have three different phenotypes. The heterozygous organism is different from both the homozygous dominant and the homozygous recessive. An example is hair curliness. So if you have curly hair, and I'll just use a big C for curly, that looks very different from the little c in straight hair. But some individuals have a big C, little c, and they have wavy hair. So that's an example of incomplete dominance. Codominance then occurs when you have two alleles expressed exactly the same amount. So you might have, so we'll use the example that we typically use, and the one example I know is blood type. So you can be type A blood, and then you have proteins that are type A proteins. So you have these things called immunoglobulin A's, okay? But I'm just going to put A means you have A blood type. Oh, not like that. If you have an A, you have type A. You can also have a B and be type B, right? Anybody who has both type A and type B proteins, they are called type AB, and they can then pass on the A or the B to their offspring. And that is codominance, because both of these proteins are expressed equally. So here we're looking at hair, okay? And so because we always use the same letter, they're using H1 to be, I believe H1 is, that's really weird the way they did it. Okay. Oh, I see. They screwed this up. This should actually be H2. Okay. So H1 is straight hair. So I'm just going to put it up here just so you know. And then I'm going to do it my way just to show you. And then H2 is curly. And then, so if you have an H1, H2, wavy, okay? That should be a VY, okay? So that's what they're showing you down here. Well, that's what they're showing you here. So if you have two parents that are heterozygous, so you have an H1, H2, so you have these wavy-haired parents, right? Then you're going to end up having one straight, two wavy, and one curly. So now I'm going to show you how I would do the same thing. Okay, so there's your toolbox from this one. I'm going to just make my own toolbox, and what I would probably do is I would use C's. So big C, curly. Big C, wavy. Little c, straight. This just gets rid of some of those extra letters, okay? So then I'm going to go over here. So what mom then would have been big C. Dad would have been little c, okay? So now we have heterozygous offspring. So I'm going to put that over here, and I'm going to put big C, big C, little c. Okay, so then again, you have big C, little c, big C, little c, right? So we still have that little c, little c is straight, and that's brown here, So 1 out of 4 will be straight. So I'm going to put 1/4 are straight. Then I have big C, little c right here and right here. And so there's 2, and we have 2 here, wavy. So 2 of 4 are wavy. And then we have 1 big C, which is curly. And again, we have curly and curly. So that's 1/4 is curly. 1/4 curly. So we still have that 1 to 2 to 1 ratio. It's just this to me makes more sense than using the H1, H2. It confuses me. So I usually tend to do it this way. You can do it either way though. All right, so let's look at some incompletely dominant disorders. The first is familial hypercholesterolemia. So familial hypercholesterolemia is a disorder that is associated with receptors in the plasma membrane that are supposed to bind cholesterol, LDL cholesterol. And so what happens is individuals that have this disorder, their proteins that typically bind cholesterol are faulty. So they don't bind the cholesterol. Cholesterol builds up in their blood, and this leads them to be very prone to things like heart attack. Obviously, they have high cholesterol, high blood pressure, and death at a young age. But it shows incomplete dominance, which means you can have the disease but have a less intense form of it. So I'm going to use H's for the disease, okay? So HH, severe, and I'm just going to use FH, okay, familial hypercholesterolemia. You can have big H, little h, and then you have intermediate FH. Or you can have little h, and be normal. So I know a lot about this disorder because my husband has the disorder and 2 of my children have the disorder. My husband's alleles are big H, little h. How do I know this? Because I'm normal, little h. So it was like doing a test cross. Had my husband been big H, all of my children would have this disorder. But what happened was I have 4 children, 2 girls, 2 boys, and 1/2 of my children actually show this disorder. So we'll just put my husband's alleles down here. We'll put my alleles up here. Okay, that does not work, does it? Sorry. We'll go like that. Big H, little h, there we go. So half of my children have the disease and half of them don't. Now, there was always that chance that all of my children, even though he is heterozygous, could have had the disease, or all of my children could have not, because again, it's just dependent on which of the chromosomes gets passed to the sperm, in this case, that fertilized the egg. But I have 2 children, a daughter and a son, that have normal cholesterol levels. And I have 2 children, a daughter and a son, that have, that have the disease. So that tells me that this disease is, or that my husband is heterozygous and not homozygous dominant for the disorder. Another disease that I believe your text states is an autosomal recessive disease is what I say incompletely dominant is sickle cell disease. And so this is a rare disease in which red blood cells are mutated. So the protein for hemoglobin differs by one single amino acid. There's a point mutation in the genetic code for the hemoglobin, and that causes the hemoglobin to change shape, which causes the red blood cells to change shape. So typical typical red blood cells are going to be circular. They kind of have this it almost looks like a donut shape. They don't have a hole, but they have this indentation in the middle. In a sickle cell characteristic, you're going to have blood cells that look like that. So they look like little sharp sickles. And what happens is these blood cells, because of their shape, they can clog blood vessels, specifically your really small blood vessels, capillaries, and then they cause an inability to move blood around. They can also cause dangerous bleeding out because they can rip blood vessels open. So they cause poor circulation. They don't carry oxygen like they're supposed to, and you're more prone to infections. And like I said, internal bleeding. So it's very dangerous. So this is what the sickle shaped cells look like versus normal blood cells. Like I said, there's that indentation in our normal blood right here, but sickle shaped cells look very different. And so why do I call this an incompletely dominant disorder? I'm going to just draw over here. If you have a big S, then you have normal blood, okay? If you have little s, you have sickle cell anemia. But if you have a big S and a little s, you have both types of blood cells. You have regular blood and sickle-shaped blood. So you have both. And so you have what we typically call sickle cell trait. And sickle cell trait can lead to things like you might have be more prone to anemia. You may have some problems with blood flow, so poor circulation. But you're going to be healthier on average than someone who has sickle cell disease, the full sickle cell anemia. You will still likely need treatment, but you can survive. Whereas sickle cell disease is very dangerous and people die from it. What's interesting, what I find interesting in this disease, is that where it is most common, since this is a disease that will kill you, typically you would assume that the disease would die out. But because of where it's located, it's most common in Africa. And Africa also is where we see malaria. Oh no. So, malaria. So, what's cool about this disease or this disorder is that the protozoan that causes malaria, so protozoan that causes malaria can only, ARIA, that's an I, can only infect normal blood. So if you have sickle cell disease, you can't be infected by the protozoan. But even if you have sickle cell trait, so right here, if you are intermediate, now you still might have a few of those characteristics of sickle cell, but you're not likely to die. But because you have some blood cells that are going to be sickle shaped, you're going to be still immune to malaria. The protozoan will not survive in your blood. How cool is that, right? So this disorder that can kill, because it is an incompletely dominant disorder and it shows differences between the homozygous dominant and the heterozygous, you are more likely to survive in that heterozygous state. All right, I am going to stop here. I believe that we're going to get into, multiple allele inheritance in the next video. So I'll stop here and I'll talk to you in a little bit. Bye.